Okay, now we get into DNA structure. So there's two parts of DNA we really focused on this unit. We focused on DNA structure, and then we focused on DNA analysis. So DNA structure is up first. Couple key things that you need to know about DNA. DNA is two-sided. So if you look on my image here, you have a blue side, and then you have a red side. So DNA is double-sided. When they get together, they form a twisted ladder structure. So you can see these are the rungs of the ladder. And then you have it, when they connect, they make the DNA twist in a spiral. We call this structure, the twisted ladder, a double helix. The helix is the twisted part of it, where it twists, and then double means it's double-sided. Now, key thing is on your DNA, the connecting parts here are made up of your bases. Now, each one of these rungs is a pair of nucleotides. Okay, it's a pair of nucleotides called a base pair. Now, a nucleotide has three parts. It has a phosphate, a sugar, and a base. My kids, remember what I taught you. You have your phosphate on your fist, your sugar on your shoulder, and a base that connects. So, in this backbone, if we look at this one right here, we have a phosphate and a sugar in the backbone, and then a base that connects. Then on this side, we have a phosphate and a sugar on the backbone and a base that connects. So DNA, we said, is double-sided. Each side has nucleotides and they base pair in the middle. So we end up with phosphates and sugars make up our backbones. So if you look at the big model of DNA in our room, we have our red and white structure, or our red and yellow structures are our phosphates, and then the gray ones are our sugars. And those make up the backbone, and then connecting in the middle is our bases. Now, DNA stands for deoxyribonucleic acid. That's where you get the DNA, and it is made up of our nucleotides. So remember, each nucleotide has a phosphate, sugar, and a base, and we connect two nucleotides to form your double-stranded DNA. The nucleotide is also called the monomer of DNA. It's just the repeating unit. So mono means one, so it's a single unit of DNA. We got lots of monomers together, makes a full side of DNA. Now, there are 4 different nucleotides. Remember, with a nucleotide, we have a phosphate, a sugar, and a base. Phosphate, sugar, and a base. Now, no matter what, you always have a phosphate and a sugar in your nucleotide, but it's your base that really identifies which nucleotide it is. So there are 4 possible bases. We have A, which is adenine, T is thymine, C is cytosine, and G is guanine. A, T, C, and G. So a nucleotide is a phosphate, a sugar, and a base. And the base is what identifies which nucleotide we are talking about. So there's a couple structures here. So on this image, I show you 2 sides of DNA. This is one side of the DNA, and this is your other side of DNA. So we have 2 sides of DNA. We said that we have phosphates go down one side, and they are we have nucleotides that go down one side. We have them base paired with another set of nucleotides. So we have a phosphate, a sugar, and a base. This is one nucleotide right here. We have a phosphate over here, a sugar, and a base. This is a second nucleotide, and they are base paired together. Now, we said on the previous slide, the sugars and the phosphate make up our backbone. So we would have lines of sugars and phosphate if I had more nucleotides. And here we would have lines of sugars and phosphates. Bases connect in the middle. They are connected together by what we call hydrogen bonds. Okay, hydrogen bonds are what hold those together. So again, you have phosphate, sugar, and a base. Phosphate, sugar, and a base. They connect together by hydrogen bonds. Okay, so identifying nucleotides. You need to be able to identify a nucleotide based on its structure. Now, there's 2 ways to do this. We look at the number of hydrogen bonds, or we compare the number of carbon rings. Okay, 2 ways to do this. So the first way, the number of hydrogen bonds. If they have 2 hydrogen bonds between them, it is either A or T, adenine or thymine. If they have 3 hydrogen bonds between them, they are either cytosine or guanine. Hold up. There we go. Sorry, I missed a G. It's either cytosine or guanine. So 3 hydrogen bonds is either cytosine or guanine. 2 hydrogen bonds, it's either adenine or thymine. The way I remember that is I think T is 2. So 2 hydrogen bonds has to be thymine. And I know that A and T always base pair together, no matter what. A will always bond with T. T will always bond with A. On the flip side, C will always bond with G, and G will always bond with C. So if I remember that T has 2 hydrogen bonds, and T always bonds with A, then they're going to have 2 hydrogen bonds between them. If C has 3 hydrogen bonds, it will always bond those 3 with our G's. So that's one way to identify, looking at the hydrogen bonds. Then we look at the carbon rings. If they have 2 carbon rings, they are a purine, which means they are either A or G. Okay, the purines, the way I told you to remember it is angels and God are pure. So A and G are purines. A and G are purines. Angels and God are pure. It's the easiest way that I know to remember them. And I know they have 2 carbon rings because I think 2 halos. Angels and God are pure, 2 halos, 2 carbon rings. Now your pyrimidines, so your leftovers, pyrimidines only have 1 carbon ring, and your options are C and T. So let's practice. Okay, so if I'm looking at this, here's what I look for. First thing is I'm going to look at my hydrogen bonds. So I know this is a phosphate, a sugar, and a base. Phosphate, sugar, base, I know my hydrogen bonds are going to connect over here. So this one has 2 hydrogen bonds. So 2 hydrogen bonds. I remember thymine is 2. So thymine and adenine have 2 hydrogen bonds. So my options are T or A. Okay, that's where I'm at now, just looking at the hydrogen bonds. Then I look at the number of carbon rings. Well, this is a phosphate, a sugar, and a base. When you look at your carbon rings, you only look at the base. So here I have 1 carbon ring and I have a 2nd carbon ring. Now I remember my saying that says that angels and God are pure and they have 2 halos, so they have 2 carbon rings. So A and G are my purines. So this nucleotide must be A, adenine. It has 2 hydrogen bonds and 2 carbon rings, so it's got to be adenine. Okay, so the key things to take away is that A always bonds with T and C always bonds with G. Always. A always bonds with T, T always bonds with A. C always bonds with G, and G always bonds with C. Now, A is a purine, T is a pyrimidine. So a purine and a pyrimidine always bond together. C is a pyrimidine, G is a purine, and C and G, so a purine and a pyrimidine, always bond together. Now, what identifies you and your DNA is your sequence of nucleotides. So if you look at your sequence of nucleotides, it's going to be different than my sequence of nucleotides. If you look at compared to Miss Alexander's nucleotides, they're going to be different. So the sequence of nucleotides is what makes you unique. It's what makes you an individual. So that is a key thing to remember. Don't forget, A bonds with T, C bonds with G. Your A and G are purines because they are angels and God are pure and they have 2 carbon rings. And then A and T have 2 hydrogen bonds between them because T stands for 2. Okay, you got to remember that. You got to be able to identify your nucleotides.